If you are intellectually curious, well, this is a deep dive you really need to hear. The future of drug discovery is definitely here, and it's being built on language models. But these aren't reading text, they're reading biology itself.
Huh.
Today, we're diving into what feels like a major milestone in AI and health. How a model, part of the Gemma family, didn't just crunch data. It actually discovered a totally novel, testable pathway for cancer therapy. A pathway nobody knew about before.
Okay, so let's unpack this a bit. The tool that made this leap. It's called C2S Scale 27B. Think of it as maybe a biological search engine, a huge 27 billion parameter foundation model built just for understanding single-cell data.
And what's genuinely, I think, groundbreaking here isn't just the scale, it's the nature of the discovery.
Sure.
This AI, it moved way past just finding correlations in data we already had. It generated a truly verifiable hypothesis about complex cell behavior. And, researchers then took that prediction and confirmed it with experimental validation in living cells.
Damn.
So the AI didn't just summarize things, it kind of invented a step in biology we hadn't seen. Right. And that invention tackles one of the really tough problems in cancer immunotherapy. How do you take these so-called cold tumors, the ones the immune system basically ignores?
Invisible.
Yeah, invisible, and turn them hot, make them stand out? The key strategy seems to be forcing something called antigen presentation, like making the tumor put up a big flag saying, hey, I'm a threat, attack me.
Exactly. That's the goal. So the task they gave this C2S Scale 27B model was super specific. Find a drug that acts like a conditional amplifier.
Conditional amplifier, meaning?
Meaning it had to boost that immune signal, that flag waving, only when a specific immune context was already there. Specifically, low levels of a key immune protein, interferon, had to be present just a little bit.
Okay, so not just any boost, but a boost under very specific circumstances.
Precisely. And that specific requirement, that need for conditional reasoning, that's why you needed such a large model. A smaller one just couldn't handle it. It's like trying to find a key that only works if the door's already cracked open a tiny bit. You need that massive scale, 27 billion parameters, to sift through all of the biological noise and spot that really specific, context-dependent effect. Smaller models, they get lost.
Okay, that makes sense. So how did they make sure it wasn't just finding, random correlations or false positives? The sources talk about this clever mechanism, a dual context virtual screen.
Oh, yes.
So you had, one, this immune context positive setting using real patient samples that had that low interferon level, and two, an immune context neutral setting using just isolated cell line data. Why run it in two different contexts like that? What was the point?
Well, it's the ultimate filter, isn't it? You want to be sure the drug works because of that specific immune environment, not just because it's, say, generally toxic to cells or something generic.
Right, isolate the conditional effect.
Right, exactly. And running that dual screen allowed the model to spot this striking difference, this huge context split for one particular compound. A kinase CK2 inhibitor called somatassertib, also known as CX-4945.
Somatassertib. Okay, I know CK2 inhibitors are fairly well studied. They flip certain cellular switches. But what was the AI's prediction here? What was the novel part?
So the model predicted it would strongly increase antigen presentation, that flag waving we talked about, but only in that special immune context positive setting. With the low interferon, specifically, it predicted it would enhance MHCI expression. MHCI is basically the cell's display window showing the immune system what's going on inside.
And this was new.
It's totally new for this drug. Inhibiting CK2, the somatassertib, what hadn't been reported before to boost MHCI or antigen presentation like this. The AI wasn't just retrieving known facts, it was predicting a new function.
A genuine prediction. And they didn't just leave it there, right? They took it to the lab bench.
They did. They tested it in the human neuroendocrine cell models, crucially, models the AI had never seen during its training.
That's important. A clean test.
Absolutely. And what happened when they treated these cells with both somatassertib and that low dose of interferon?
What were the results?
They saw a really marked synergistic amplification of antigen presentation. We're talking roughly a 50% increase.
Wow, 50%. That sounds significant.
Oh, it's highly significant.
Yeah.
That kind of boost could potentially make a previously cold, invisible tumor much more visible and vulnerable to an immune attack.
So if we zoom out a bit, what's the bigger picture here? What does this tell us?
Well, I think this result, this experimentally validated finding, It provides a kind of blueprint for a new way of doing biological discovery.
Yeah.
It really demonstrates that if you follow the scaling laws, if you build these larger, more capable foundation models for biology, you can run these incredibly high-throughput virtual screens and find really complex, context-conditioned biology, things we might miss otherwise.
So it's not just a potential drug lead.
No, it's more than that. It's an experimentally validated lead for potentially developing new combination therapies using this drug alongside maybe interferon or other immunotherapies, and it shows the power of scaled AI in this space.
That's incredible. And for any researchers listening who want to explore this, the C2S scale 27B model itself and the related resources, they're actually available, right?
Yes, they made the preprint available on BioRockSorv. You can find the model on Hugging Face, and the code is up on GitHub, so the community can build on this.
Excellent. Good to know. And a quick note that this deep dive was powered by embersilk.com. Embersilk is the answer for all your AI education and integration needs. Check them out at embersilk.com.
You know, thinking about this whole thing, it really raises a profound question for the future, doesn't it? If AI can develop these entirely new capabilities like high-level conditional reasoning and genuine hypothesis generation, specifically in biology,
Yeah.
well, how soon will it be before we rely on it to generate the majority of our future clinical hypotheses instead of just using it to process the data after the fact?
That's definitely something to think about. Where does human intuition fit in versus AI-driven discovery? A fascinating question indeed. Well, if you enjoyed this dive into the absolute cutting edge of AI and health, please do remember to subscribe to the podcast and maybe give us a 5-star review. It really does help us bring more deep dives like this to you. Thanks for listening.